Oh my goodness. Yeah, there is so much fun. Summer.
Yeah, it's like, so everybody.
Although we like sitting in seats, I feel like
You're from like, so you home to Arizona?
Yes, that is.
It's hotter than freaking heck. I didn't think it was that. Where you're at, or?
I feel like when you wear your mask,
I guess that's some Arizona. I think it's Flagstaff. Oh yeah. It's like 20 minutes from Louisiana. It was full on like swamps, like there was another scout camp. We had lived with like acres in it, and like catfish and so. Oh, I love it. Like these Texas guys, good infrastructure, but it was still really nice and good food.
Welcome, everyone. Welcome. Let's go ahead and get started. I'd like to begin with prayer. Is there someone willing to provide a prayer for our class today? Please.
Dear Heavenly Father, we're grateful for this day, and we're grateful that we have the opportunity to meet together this day and learn about biochemistry. And we ask that God please help us to have the Spirit to be with us as we learn today, that we can retain and remember these principles. And we're so grateful that we have the opportunity to gain an education, and we ask that God please help us to remember Christ in everything that we do. And please help us as we go throughout our semester, that we can all stay healthy and well. And we love you and say these things in the name of Jesus Christ. Amen.
Amen. Thank you. Thank you very much. So I am going to distribute the quiz again and this handout on mass spectrometry. I'm guessing that most of you have already started on the quiz and may not need a hard copy, but nevertheless, I will distribute those. So, are there questions that you have at this point? Yes.
I've just been like rewatching the lectures. So basically everything we've been talking about the last couple lectures is just like giving a crude separation and then a more specific separation, and it's all about like isolating products.
That's correct. Yeah, so the textbook talks about separations, but doesn't give you enough information to actually understand how they work or why you would use one versus another. So my intent is to give you enough information to allow you to make rational choices if you were called upon to do so in a quiz or exam, which you will be. And the intent of this is to help you understand how one then isolates proteins or other biological molecules. And our discussion about mass spectrometry, which is touched on very superficially in the text as well, is to help you understand how proteins are identified currently. virtually all protein identification and most of the other characterizations of lipid molecules, sugars, metabolites are all conducted using mass spectrometry. Nucleic acids, so RNA, DNA, they have their own specialized techniques. These are very robust. Those are talked about in our book, but not talked about in this class. They're talked about in Chem 482 and in a variety of classes taught in life sciences. So that's sort of where we're heading. Other questions that you have? All right, then we're going to talk today then about how one actually goes about identifying proteins using mass spectrometry. It's not as simple as one might anticipate. These techniques in some ways are relatively elegant, but they're also relatively complicated. We won't, hopefully I'll give you enough information to allow you to understand in general how these processes occur. And then we're going to move on. I'm going to go back to a slide here. Okay, here's where, let's see, I do the stop sharing. Let's see, I go here. All right. All right, so we had sort of ended with this slide, and we had talked about the various types of mass spectrometers that are available commercially. There are other kinds that are used in research, but in terms of what one will encounter in a pharmaceutical laboratory, in a hospital laboratory, if you're at a large tertiary hospital, these would be the types of instruments available. You'll remember that the quadrupole was the least sensitive and the least accurate. I mean, it's still very, very good. As mentioned, it's probably a well-tuned, well-cleaned instrument is capable of distinguishing mass differences as little as 1/1000th of a mass unit, 0.001. So if you're talking about a protein that's 50,000 Daltons, 0.001 beyond that is pretty accurate. The time of flight instruments, the ones that are commercially available now, do about 10 times better, both in terms of sensitivity, how little you can measure, and also 10 times better in terms of accuracy. So now we're able to distinguish molecular species that differ by as little as 1/10,000th of a mass unit. And the Orbitrap is currently the best commercial instrument that is available and does even better. So it is even more sensitive and more accurate, down to, let's say, even 100,000th of a mass unit. So what does this mean? What does this look like? So let's say you take you grow cells and you lyse those cells and you're looking at the cytosol. If you were to run the cytosol, even using something like electrospray ionization, where you spread things out through a separation column prior to introduction onto the mass spectrometer, you're going to see maybe a couple hundred peaks. That is, you can differentiate maybe 200 different compounds. If you use a time of flight instrument, now you're talking about 2,000 to 3,000 peaks. So you're able to keep track of 10 times more peaks. And on an Orbitrap, you can monitor as many as 20 to 30,000 peaks. So you're able to keep track of, monitor, measure, investigate, interrogate, whatever you want to say about that number of peaks. So it's a very substantial number. Now, data management analyses become more complicated as you have more peaks to look at. On the other hand, the coverage of what's actually present in the cytosol of a cell or the serum of an individual goes up proportionally as well. Okay. Now, before I leave this slide, I want to point out one thing. So one might think that quadrupole instruments are really of limited value, but this is a little bit misleading. One of the features of a quadrupole instrument is that it is non-destructive. And we're going to talk about, as we talk about amino acid sequencing, we're going to need the ability to have coupled together two mass spectrometers, a tandem mass spectrometer. And in the first, you use the first mass spectrometer to select one particular ion, one particular molecule, and it's going to be moved into a compartment within the mass spectrometer called a collision cell. It's going to be collided with neutral gases, and fragments are going to be produced. We'll talk about this in more detail in a second mass spectrometer. Now, the second mass spectrometer can be another quadrupole time-of-flight instrument, or it can be an Orbitrap, but it is absolutely necessary to have a quadrupole as the first dimension of this two-dimensional mass spectrometer, this tandem mass spectrometer, because an Orbitrap or a time-of-flight drift tube will not allow you to select and move an ion to another part of the mass spectrometer. They are destructive. You're measuring mass very accurately, but you cannot use that part, that time-of-flight tube, to move an ion to elsewhere in the mass spectrometer. Okay, so that's a bit of backdrop. Let's move ahead then. Let's talk about the situation where you might use protein fingerprinting, okay? So I'm showing here two-dimensional gels. You'll recall that the top of these are separated initially based on their isoelectric points. So let me go here, and so up here, if you look down at the bottom, you can see, if I can find my dot here, you can see that we have pH, okay? We have a series of pHs for both gels. These are equivalent, done under equivalent conditions. So we're going from lower to higher pH. And we have this in a tube. The tube has a gel in it. We apply a voltage. Proteins separate then based on their isoelectric point. You'll notice that we will be, based on this application, that proteins with a isoelectric point below about 4 or above about 7.5 are not going to show up. They're going to be excluded. Now, in the second dimension, so this gel is, this isoelectric focusing step then results in a gel that has proteins partitioned based on their isoelectric point. This is overlaid on a sodium dodecyl sulfate, and we run this dimension. So you'll notice that we see numbers here that are indicative or proportional to the molecular weight of these species. And so we go from the top, where you have large molecules that can't negotiate the gel very easily, to those that are quite small and can work their way through the gel very rapidly. And the consequence of having two dimensions is that you spread things out now based on two independent physical chemical properties. In one case, sort of the electrochemical charge on the protein, the second based on the size of the protein, and as a consequence, you are able to visualize then dozens, maybe a few hundred spots, each of these representing a protein. Yes, go ahead.
I was just wondering,
Can you speak up? So.
yeah, what's on the x-axis, like your isoelectric waves, does that vary based on like what molecule you would be looking at?
Yes. So the question is, why the particular pHs that we have here. So yes, you as a scientist would have to make some sort of rational choice. It turns out that the majority of proteins are going to fall within this pI range, and that's probably why the investigators chose this. But you are losing some information by limiting the pH range of your amphiolites. Now, if you look at this gel, if you study these two, you'll see that there are differences. Now, so some of these, yes, the gel's not exactly the same between the two, but you get the sense that, yeah, they're largely the same. But you'll see that some spots are missing, present in one and missing in another. So I don't know, so if I look over here, I see one right here, a little spot that doesn't appear on the right, okay? So I have a tumor as shown on the right that has been homogenized and run out on these gels. And on the left, I have presumably normal tissue adjoining the tumor, same tissue, same organ, same patient, but normal looking tissue. And so we're now making a comparison between these two. What you can do is I can come and I can take this spot here that I've circled on the left, and I literally take a scalpel and I excise that dot out of the gel. And the intent is I want to identify that protein. I want to know why this protein is present or increased in normal tissue, but lost in tumor. Okay, so this is the question. What is the protein? What may be its function? All right. Now, yes?
You can't run both of those dimensions at the same time. One has to be done before the other. Right?
That is correct. Yes, so the point made is that this actually represents two sets of experiments. You do the isoelectric focusing, you get a little tube gel and you overlay that on a cast gel made up of polyacrylamide, and you run SDS Page thereafter. So two separate experiments that allow you to do this. Yes?
So just to clarify that part, you're literally just going to take the gel out of the tube, put it on top of this polyacrylamide, and then run it.
Yeah, so it sounds crazy, but it works. So the tube is laid flush on top of the tube gel is laid flush on top of the polyacrylamide gel, and you add some SDS to the solution so that the SDS has an opportunity to interact with those proteins, and then you electrophoretically move the materials from the tube gel onto the polyacrylamide gel and on through the experiment. Okay.
There's another question.
Yeah.
Will the denaturing of a protein by SDS Page change the MS results?
Good question. So will denaturing the protein, as happens with treatment with sodium dodecyl sulfate and boiling and so forth, change the mass spectrum of the protein? And the answer is probably not. in the vast majority of cases, it will not affect it. These techniques, I mean, even though you're unfolding the protein, you're not changing the composition of the protein. You're not fragmenting it in the sense. So you should see the mass spectrum should be the same. All right, so here's the first of the two techniques used for identification of proteins. This goes by, it is called sometimes protein fingerprinting. Sometimes it's called peptide fingerprinting. The concept here is you take a purified protein. This is typically how it's done. So remember now our two two-dimensional gels, we excise a spot, we recover that protein. We feel that it's fairly pure because there was no spot, comparable spot on the other tissue. We then take that and we put it in a tube and we mush up the gel so that the protein can escape, and we add an enzyme. This is a proteolytic enzyme. It's going to break the protein. This particular enzyme, the one that is used almost exclusively in this type of approach, is an enzyme called trypsin. And it selectively cuts proteins in pieces at arginines and lysines. So it's not random. It is systematic. It's going to cut proteins at selected places. Now, each protein has its own amino acid sequence. Consequently, as you treat each of these proteins with trypsin, you will produce a set of trypsin digest fragments or peptides that are unique, a unique set. Now, some pieces may be the same if you have two closely related proteins, but because the amino acids or sequences are different for every protein, you will end up with different trypsin digest fragments produced. In fact, you have a unique characteristic set of trypsin digest fragments for any given protein. So in effect, you have sort of a molecular fingerprint for that protein that represents now these pieces produced by trypsin digestion. These in turn, so we take our protein here, it's submitted to trypsin digest fragmentation. So we cut it in pieces at tryptophanes and, not tryptophanes, at lysines and arginines. And then we are going to study those pieces. We're going to actually now run them on a mass spectrometer. We're looking just at the masses of those pieces. We're not trying to amino acid sequence them. We're just, how much do those pieces weigh? What is their molecular weight? And so that's what we are going to find out using mass spectrometry. What then happens is you, based on the species, you will use a very sophisticated database that looks at all of the genes in a species, so let's say the human, and it will look at all of the predicted proteins produced from those genes. So let's say there are 30,000 genes or something like that, and 30,000 gene products, 30,000 proteins. This can all be done in silico. This is all done by computation. And then you can predict what the peptide, the trypsin digest fragments would be for each one of those 30,000 proteins. Now, if we were having to do this by hand, it would be a long process, but this is done very quickly, very simply by computers. So we now have this database with all predicted trypsin digest fragments for all proteins in an entire species. So we run our trypsin digest fragments, and we're now going to compare them to this hypothetical set of trypsin digest fragments. The fragments will differ based on what the proteins are. And you only have to get 5 or 6 matches between your experimental data and this database to know exactly what protein you have. So it's very, very sophisticated and it's very robust. You'll notice here that some of these are modified. So these are post-translational modification. This one represents carbohydrate addition. This one, phosphate added to a serine, threonine, perhaps a tyrosine. So these pieces will not match the theoretical database. When we look at genes, they don't describe future post-translational modifications. They don't have that ability. So we'll need to have enough fragments that we can still have 5 or 6 unmodified protein digest fragments that match with our database. So we may lose half of our species because they are post-translationally modified. Yes?
Does the database come from like when they did the human genome all the way through?
Yep. Yep. Yeah, and the chicken genome and the zebrafish genome. You know, not all species have this information available, but most, but humans do, and most laboratory animals do, and a lot of agricultural animals do. And so the fruit fly has been studied exhaustively, and zebrafish and mice and rats and so forth. Yes. Yes.
So in our hypothetical situation, we're talking about a tumor, correct?
Yeah.
A tumor protein. So if that was the case, we're obviously in a hurry to identify that protein. Is this process relatively quick, or does it take quite a bit of time?
So once you have the protein pure, that is, after you've run the gels and you see a spot that differs between normal and diseased tissue, you can excise it, do the trypsin digestion, apply this to a mass spectrometer, and this is where MALDI can come into play because you have a purified protein. You can take the trypsin digest fragments, mix them with your matrix molecule, spot them on a MALDI plate, irradiate them with laser light, and the data, the mass spectrum is almost instantaneous. You can have a mass spectrum within a minute or two of that pulse of laser light. This comparison to the database, you actually send this off. There are actually sites with tools available, and modern mass spectrometers software actually will automatically send it off. And so in the matter of a day, I mean, you can get this comparison in a matter of minutes. And so within a day, you can pretty much do the whole experiment. Now, two-dimensional gel electrophoresis is a black art. And so being able to do these gels so that they're comparable takes somebody really skilled in the art. It's not anybody who can walk in a laboratory and do 2D gel electrophoresis. But nevertheless, the concept is once you have the protein, you can get the identity of the protein and even the gene that produces it or codes for it in a matter of a few hours. Okay, so this is sort of what the data might look like. The ones, yes?
Do they use this for diagnosis or more for research?
Well, it could be used for both, but currently most of this is for research. Now, you could potentially look at certain gene products as a way of identifying what type of tumor you might have. You could potentially use this to look for other kinds of diseases that involve modifications of proteins. So let's say, it turns out that in many cases, so if, for example, we could look at hemoglobin from a sickle cell anemia patient. They have a protein, the hemoglobin has a protein sequence that differs from that of a non-cycler, okay? So this could be determined fairly quickly using mass spectrometry if you had the two sources of hemoglobin, a normal individual and a sickle cell individual. You can also do this genetically. I mean, you can also look at DNA and know that this person carries the sickle trait or has two copies of the allele. So it may not be the fastest way of getting the information you want, but it could be used in that way. Most of this is probably for understanding the physiology or pathophysiology of diseases in an effort to eventually treat the underlying problem. So, okay, so here, yes, go ahead.
And then somebody asked if you often run the 2D electrophoresis. Like, do you know how to do that?
Do I personally know how to do it? I have done it. It has been done in my laboratory I had a grad student who did it. And he struggled. I mean, he was there doing these for a year before he was able to get useful data out of doing 2D gel electrophoresis. The problem that he was dealing with was, does the diet of a fruit fly change its protein composition? So there's something called the hamburger hypothesis. That is to say that you can see changes in the protein composition of an individual if they've had a hamburger, let's say, versus, I don't know, a salad. Yeah, okay. So, it turns out that diet didn't have that big of an effect, except for certain kinds of specific foods. So fruit flies like ethanol. In fact, they require ethanol to live. Anyway, he found out that ethanol was important, which was already known, but we could see differences in the protein composition. So, okay, so here's these now represent trypsin digest fragments. Okay, they are these digest fragments are spotted. Let's say in this case on a MALDI plate and irradiated, and you get this array of peaks. And if you you have software on the instrument itself that will actually list these out, so you can see a list of the various masses of these fragments, okay? And you can, this is the actual interface with the database. You can tell it which species that you're dealing with. If it's unknown, you can look at all the currently available species that are in the database. And you get this kind of result back. So you can see on the left here, the actual masses. So this is the experimental here. Here's the theoretical one. This is generated from genomic information and predictions of the trypsin cleavage sites, producing a set of trypsin digest fragments. So we have the experimental and theoretical. Over here on the, based on the genome, the known genes, and we see over here the sequences represented by these different masses. Now, let me emphasize that this is not sequencing these. It's depending upon the base sequences of those genes in the database to predict what the amino acid sequence would be for theoretical fragments and consequently for experimental work as well. And down at the bottom, you can see that you can, where you have highlighted letters down there. These represent amino acids. That represents coverage. That is, there is a fragment that overlays that part of the protein. Now this identifies this. Okay, so up at the top here, we see that this is myoglobin. And so here's the identification of the protein. Down at the bottom is based on genomic data, is the exact amino acid sequence. This is not based on mass spec data. This is based on base sequences in the DNA producing products. Okay, yes.
So I noticed that the experimental is always just barely lower than theoretical. Is that significant at all? Or
So these, these are probably measured on a quadrupole. So anything from a practical point of view within 100th is probably okay. You'll notice that theoretical has a lot more decimal points because you know exactly how many carbons, hydrogens, oxygens, nitrogens there are, and you know those atomic weights quite accurately. So, but small differences may happen. You know, your instrument may hiccup, and so you may get small differences. Systematic differences, it might be the tuning of the instrument. It may be miscalibrated slightly. So, yeah. Yes?
So on the experimental side on the position, it has a start and end. Is that where the trypsin's cutting this protein?
Yes.
And then you put them all together to get that sequence at the bottom?
Well, the sequence at the bottom, the entire sequence is determined based on base pairs. And so the coding provided by the gene. So you have a start codon, a stop codon that's recognizable. And you look at the base sequence and say, okay, we're going to have an alanine, oh, leucine, now this and that. So the entire sequence at the bottom is based on genetic information, where you see highlighted letters that are shaded. That indicates that you have conformation with with these fragments. Okay, so. Okay, let's try to move on here. We've got to talk about one more method. So the second way of approaching protein identification. Okay, so this whole conversation is about how we identify proteins in our day and age, and that is done with mass spectrometry. But there are, in fact, two ways of going about this. The first was the protein or peptide fingerprinting. The second of these deals with actual amino acid sequencing. And let me state at the beginning and emphasize that this requires a tandem mass spectrometer. A tandem mass spectrometer actually has two separate mass spectrometers, and it has between those two something called a collision cell. And the idea is that we use the first mass detector, the first mass analyzer. We're going to use it to select one particular molecule, molecular species, molecular ion, to undergo fragmentation in our collision cell. Okay? So we can run a full mass spectrum. We can see dozens or hundreds of species using our mass spectrometer. And so let's say we're doing an experiment that's not unlike the 2D gel electrophoretic experiment. What we're going to do is we're going to take, let's say, a homogenate of normal tissue, a homogenate of cancerous tissue, and we're going to run them on our mass spectrometer. We're just going to look at all the peaks that are visible that show up in our mass spectrum. But let's say that in that process, we see a peak that's present in tumor that is not present in normal tissue. Okay, so to understand this, we're doing comparative. We have to run two experiments, one using normal tissue, one using abnormal tissue. We go through the whole process. In the end, we we submit both of them one at a time to the mass spectrometer. If we find species that differ either in presence or absence or in intensity between our normal and our disease tissue, and this seems to be a consistent pattern, then we become very interested in wanting to know what that particular protein or molecular species is. So again, we can use our tandem mass spectrometer to survey all of the masses that can be detected in our specimen. But to do amino acid sequencing, we're going to just select one of these. This one that shows up in tumor tissue but doesn't show up in normal tissue, as an example. Okay. Now, we are going to need a tandem mass spectrometer to be able to carry out amino acid sequencing. The first part of this, the first mass detector that we're going to have must be a quadrupole. Why? Does anyone remember something about a quadrupole that was unique? Yes? Non-destructive. You can't put a time of flight in at the beginning. You can't put a orbit trap at the beginning. You have to have a quadrupole to be able to move an ion into the collision cell. Now, the presence of a quadrupole imposes a limitation. You can only measure mass to charge ratios of 4,000 or less. And without going into all the details, suffice it to say that this is a fairly substantial limitation. It means that you're not going to be able to measure full-length proteins, large proteins, very often. You may have to cut them up with trypsin, or you may have to focus on smaller species, because the quadrupole cannot handle a large mass to charge ratio. So, okay. So let's say you have 1,000 different molecules that you see when you conduct a full mass spectrum of your tumor homogenate. One of these is found only in tumor tissue. That's the one that you're going to select, and you're going to move it into this collision cell. And in the collision cell, in fact, it's accelerated through the collision cell. In the collision cell, there's a small amount of typically nitrogen gas, sometimes argon gas. Nitrogen's cheaper, so most people use nitrogen. There's just enough nitrogen or argon present to, on average, produce one collision for the ion that is moving through here. Okay? Now, some will go through and not collide. There'll be a small amount, but the majority will go through and will collide with a nitrogen or an argon gas molecule, and it will produce a single break in that protein or peptide. The acceleration energy and the the mass of these gas molecules is such that we almost exclusively break peptide bonds. Okay? That means that, you know, I can break the peptide bond almost anywhere, well, really anywhere in this protein. It can be near the N terminus, the C terminus, right smack in the middle. In fact, it's going to arbitrarily break the peptide bonds pretty much everywhere. Usually, if you have a lot of your protein, you will see breaks for all of the peptide bonds. Not all of the peptide bonds are broken. Remember, I'm only breaking one per molecule, per ion that's moved through the cell. But in the end, I'll have some broken at the end, some broken in the middle, and I'll have this series of collision fragments that represent breakage at each peptide bond within a protein. These then enter a second mass spectrometer and they are measured simultaneously. So I will see all of these collision fragments simultaneously. Let's see if I can. So here's kind of a depiction of what's going on. I use my first mass spectrometer here as a tool to select one molecular species, which is moved into this collision cell. There, it will collide with argon, nitrogen, and it will break peptide bonds. Notice that it breaks them symmetrically. That is, you get one electron left on the nitrogen and one electron left on the carbonyl that are part of that peptide bond. This leaves me now with, another molecule will go through and it will be broken here, okay? And another will go through and only it will be broken there. So I'm going to get a series of fragments that represent breaks of just one of these peptide bonds. Let me point out, notice now that this particular fragment no longer has a positive charge on it, okay? But this fragment down here will. It should be NH3. Sorry, get this. So this should be an NH3. If we've been doing electrospray ionization, that species will be positively charged, whereas the fragment on the right will not be positively charged. What does that mean? It means that we'll only see the positively charged species in the second mass spectrum. All right, so this is a little hard to see, but you'll get now a series of peaks, and they represent the full-length peptide. Let's say this actually, you don't see the parent peak, but then you'll see a peak next to it that represents the loss of one amino acid from that end. And you'll see another peak that represents the loss of two amino acids, or the loss of a 2 amino acid piece, and then a 3 amino acid piece. Based on the differences in mass, you know what the amino acid was, okay? And consequently, then you can actually assign, as is done here, you can actually look at these and say, okay, the mass difference between this peak, the parent peak would be out here somewhere. The difference between the parent peak and this next largest peak is the correct mass for it being leucine or isoleucine. If I look at the difference here between, this peak and the next peak, I know that this is a phenylalanine, okay? So I can literally see this sequence of the peptide and know what it is. Now, if I get 6 or 8 amino acids in a row, I can submit it to this same genomic database and it will identify the protein. You only need about 7 or 8 amino acids in a row to uniquely identify the protein from which that sequence came. Some cases, the sequences are peculiar enough or unique enough that you may only need 5 or 6 amino acids. In some cases, 5 or 6 amino acids in a row could be found in any of 3 or 4 proteins. So, but 7 or 8 is almost always enough to be able to uniquely identify a protein based on its amino acid sequence. Okay. Let me, we're out of time really. Let me refer you to the handout, to this sort of additional information about mass spectrometry. I try to go through all of the considerations here in greater detail. I know some of we're going through this pretty quickly. This is all new. Some of it's not making a lot of sense at the moment, but let me refer you to that handout and to the slides. They attempt to, if we had read through them, they do try to spell out what's happening. Yes, go ahead.
Sorry if you already answered this before, but there's, so it makes sense that we would kind of not worry about those little tiny spikes right there, but there's two large ones right there that we're not thinking about.
Right. So again, your computer software is able to say, well, I'm only going to consider peaks that differ by the molecular weight of an amino acid. So it can make that judgment call for you, if you will, okay? Otherwise, if you're doing this by hand, and you can, you'd have to consider all of the mass differences and sort of say, you know, this really doesn't fit in the pattern. This doesn't, the mass differences don't conform to a particular amino acid. It doesn't make sense. Now, something that does come out of this is that you can actually identify post-translational modifications, something you can't do with protein fingerprinting. You can see something that differs by the a molecular weight difference of a sugar, you know, a 6-carbon sugar, and you sort of say, oh, I have a post-translational modification. I don't know what the sugar is. I don't know if it's glucose or fructose or mannose, but it's clear that everything beyond this has that same increase in weight, and it happens right at this serine. So I know it's actually the serine that's been modified. So you can get a lot of information using this technique. All right, sorry I can't move the story forward. Have a good weekend, be safe, and I'll try to get this up on Learning Suite for you to review. You're welcome.